Now I'm going to show you a little bit of extra experiments that we did to try to test this hypothesis, and it was to do a laser ablation. Why a laser ablation? Because a laser ablation is possibly the technique that allow us to kill cells in a most specific way, not only temporally but also spatially. And I have to remind you that this section of the leg is really small. It's just few microns. So to target that region is very difficult genetically or using just a physical perturbation. If you try to, experiment, so for example, you can use heat or you can try to cut some section of the leg. However, those experiments are very difficult to do it in in this section of the leg. And genetically, we are generally perturbing a big region or only the bristles or the half of the tarsal segment. So they are not completely allow us to test this hypothesis. In the distal region, and there is no rotation, this means that the push model was the one that it was correct. However, if we kill cells here and the comb still rotate, is because the push model was correct. So it was very straightforward. And we can do the opposite. If, and let me put it in blue, if we kill cells here, but the rotation still takes place, it means that the mechanism that it will be the pull mechanism. And if we kill cell here using laser ablation and there is no rotation, again, this will be an indication that the pull mechanism, the pull hypothesis is the one that is correct. And so we try to do laser ablation in this region, and I'm going to show you what happened. What happened, if you see here, there are pieces of the tissue that start healing. So we try to do laser ablation several times. We cut it 5 times, and, and the tissue keep healing. And while we cut also on the top of the comb, we try to make also cuts in this region. Those cuts or didn't have an effect, or they end up killing the pupae. So this technique, although it's in theory sounds very interesting, it was not able to answer this question. And another problem that we have is that our cats, unfortunately, instead of we cannot make the cuts in this area, in the early stages of the rotation, we have to make the cuts here. That is not the ideal place. And why? And I will show you why in the next slide. As the leg has a curvature, uh, the best cuts that we have, they were in this region. And unfortunately, in the regions with high curvature, the cuts are not as good. So the region that we were trying to cut, that it was the ideal to test the hypothesis, we were not experimentally able to cut it. And so far the laser ablation didn't work as well as we want. And when something doesn't work experimentally, there is not techniques so far developed to answer that question. The best way to test ideas is to use computer simulations. And we talked to a friend that he was a physicist, or there is a physicist, and he used the data that we have, the changes in apical cell area, the different uh, cell dynamics, cellular parameters that we have, and he creates simulations of the process of the rotation. And he found something very interesting. The paper was published in PLOS Computational Biology. I highly recommend you that if you're interested in this topic, that you look in, not don't read the paper, or not in the beginning if you're not a mathematician or a physicist. I recommend you that you read and go to this blog that they request us to write. uh, to make it easier for people in this research, the journal Developmental Biology asked to 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 make it to write it to make it simple for other scientists. And in that section, in that link, you can see a very easy explanation of what we did. And we use as a technique to simulate that a pod model. And many times when we work with computer simulation in biology, you can find very pretty images, but there is no math underneath. There is not an equation. Here we have an equation. Here we have all the cellular parameters, and it gave us a very good information, very interesting data that allows to understand in more detail what is happening during the rotation, and maybe for many of your projects it will be very useful. And I'm going to again summarize it using a schematic. What he found is that if you see here in our simulation, there are two regions. One region that is pink, dark, dark pink. I don't know how to define that color. And this one that is blue. Why? Because when he started creating simulations of cells that they grow at a similar rate, he found that the sexcom is not able to rotate and become straight. Yeah. So he's able to move, but they form this can shape that is hardly ever seen. Only we see it when there is a problem, a developmental process. So that was very interesting. To make a straight sexcom, he needed to make these cells expand early, while these cells at the bottom of the comb, they have to expand later during development. So he found that they should be inhomogeneous cell expansion. And this is fascinating because this is very interesting for some of the projects that we will discuss in more detail later about how the changes start changing in cell shape. All this process of pulsation seems to fit with this finding that he proposed using the computer simulation.